Good morning, everyone. Today we are going to discuss about the mechanism of humoral immunity. Humoral immunity is mediated by circulating immunoglobulins in the gamma globulin fraction of the plasma proteins. Moving on to mechanism of humoral immunity. When an antigen first enters the body, it can bind directly to the appropriate receptors on the B lymphocytes. So this will cause activation of the B lymphocytes. So the receptor present on the B lymphocytes will be an antibody. That is, when this B lymphocyte synthesize antibodies, some of the antibodies will be transported to its surface and it will be inserted on the surface so that it can act as receptors. But this will not cause the complete activation of B cells. So when the B cells bind to the antigen, there will be activation of the B cells, but for the complete activation we require interleukin 4, 5, 6 from the helper T cell, that is helper T cell Th2 subtype. So helper T cells are needed to produce the full activation of B cells and antibody formation. So the antigen is presented to the T lymphocytes at the same time because your B lymphocytes can act as antigen presenting cell, so they present the antigen to the T cells, that is helper T cells, and they bind to that antigen and activated helper T cells are formed, and this activated helper T cell will release interleukin 2 that will go and act on this activated T cell itself, that will result in the proliferation. And like we studied in cell mediated immunity, the subset of helper T cells that is to be proliferated is selected depending upon the effector mechanism that is selected for destruction of that pathogen. So if that effector mechanism was by using antibody, then the proliferation will be of Th2 subtype. Then the Th2 subtype will be proliferated and they will release interleukin 4, 5 and 6 that will stimulate B cell growth and differentiation into plasma cells and memory B cells. Plasma cells will secrete immunoglobulins into the circulation. Memory B cells will be remaining there and they get converted into effector cells on subsequent exposure to the same antigen. Effector cell means the B cells only. So it will cause much more rapid and much more potent antibody response. As I have already mentioned, immunoglobulins are secreted form of antigen binding receptors on the B cell membrane. Moving on to mechanism of action of antibodies. So they act in two ways, by direct action on the pathogen or indirectly by activation of the complement system. So first we will discuss direct action via the antibodies. Antibodies can activate invading agents in one of the several ways. So first is agglutination. Agglutination is a process in which multiple large particles with antigens on their surface, such as bacteria or red blood cells, they are bound together into a clump. So you can see RBC is having antigen on their surface. So this antibody will come and bind to this antigen and hold together, they form a clump. So this is known as agglutination. Here you can see bacteria. Bacteria is having antigen on their surface. Again this antibody will come and bind to this antigen and they form a clump. So this is agglutination. Here the antibodies are directly acting. Okay. Next is precipitation. Molecular complex of soluble antigen and antibody becomes so large that it is made insoluble and precipitate. So here you can see the antigens are soluble. They are not present on any particles like RBCs or bacteria. They are present in the blood. So into this antigen, antibodies will come and attach, and this soluble antigens are made insoluble because as they become large, because of binding with the antibody, they become large and they are made insoluble. So once they become insoluble, they precipitate, and this antigen that is bound to antibody will not act. That cannot cause any problem to your body. So that is precipitation. Then neutralization. Neutralization is a process in which antibodies will cover the antigenic sites of the antigenic agent. So again this antigen cannot act on your body. Then lysis. Lysis is a process in which antibodies are capable of directly attacking the membranes of cellular agents and thereby causing the rupture of the agent. So that is lysis. Lysis means rupture. direct action of antibodies are not strong enough to play a major role in immunity. So most of the protection is through the amplifying effects of the complement system. So what is this complement? Complements include serum and membrane bound proteins that function in both natural and acquired defense system. There are 20 to 30 proteins. They are enzyme precursors. They complement the actions of antibodies and phagocytic cells in destroying the pathogen, removing the damaged cells, and promoting inflammation. Complements are normally inactive, but they can be activated by different pathways. So they are released mainly by liver, then monocytes, macrophages, endothelial cells, epithelial cells, etc. But they will be released in an inactive form, which have to be activated by different pathways. So when activated by antigen-antibody complex, this complement components react in a specific sequence as a cascade, like your clotting mechanism. The complement cascade is a series of reactions in which the preceding components act as enzymes on the succeeding component, cleaving them into dissimilar fragments. The larger fragments join the cascade and smaller fragments are released. They possess biological effect, amplifying the defense mechanism. 3 different pathways activate the complement system. One is classic pathway, alternative or properdin pathway, mannose binding, lectin pathway. Classic pathway is initiated by antigen-antibody complex. When an antibody binds with an antigen, a specific reactive site on the constant portion of the antibody becomes uncovered or activated. So that area will go and binds with the C1 molecule of the complement system. So that results in a cascade of sequential reaction beginning with the activation of proenzyme C1. So initiation of this pathway, we need antigen-antibody binding, because antigen-antibody binding will uncover the site that is for the binding with the C1 molecule. Now C1 enzymes formed then activate successively increasing quantities of enzymes in the later stages of the system. Thus an extremely large amplified reaction happens. Multiple end products are formed, and these end products have important effects in the immune function. Now you can see this is the cascade of reaction that is happening in classic pathway. Here you can see when antigen-antibody complex happens, that results in the activation of a site for binding with the C1 complement. Now the proenzyme C1 is converted into active C1. Now this C1 will act as an enzyme for this reaction. Now we are getting C42 and C4A. So one is a larger fragment and one is a smaller fragment. So larger fragment joins the cascade and smaller fragment is released. So like this larger fragment acts as an enzyme for the next reaction, and again this larger fragment acts as an enzyme for this reaction, and this larger fragment acts as an enzyme for the next reaction, and this reaction continues, and finally we get a large complex, and that causes lysis of the target cell and causes destruction of the pathogen. So you can see these are the smaller fragments or byproducts. They have definite functions in the immune system. You can see C4A, C3A and C5A. They are helping in activation of mast cells and basophils. C3B is helping in opsonization of bacteria, that is coating the bacteria so that it is easy for the macrophages to phagocytose them. Then C5A helps in chemotaxis of white blood cells. Chemotaxis is process by which the white blood cells are attracted towards the pathogen. So this is the cascade of reaction that is happening in classic pathway, and for that we need antigen-antibody binding, because on antigen-antibody binding that will result in uncovering of an active site which is meant for binding to the C1 component of the complement system. So now we will see what are the functions of the byproducts of the classic pathway. Opsonization and phagocytosis. C3b activates phagocytosis by both neutrophils and macrophages. These cells engulf bacteria to which antigen antibody complexes are attached. So this process is called opsonization. C5b6789 is also known as cytolytic complex or membrane attack complex that has a direct effect of rupturing the cell membranes of bacteria or other invading organism, resulting in the lysis of them. Now we'll see the agglutination. The complement products change the surface of the invading organism, causing them to adhere to one another, promoting agglutination. Then neutralization of viruses. The complement products or complement enzymes, they can attack the structures of some viruses and make them non-virulent. Chemotaxis. C5a initiates chemotaxis of neutrophils and macrophages. This causes large number of these phagocytes to migrate into the site of inflammation. Activation of mast cells and basophil. C3a, C4a, C5a mediate this function. On activation, they release histamine, heparin, and several other substances into the local fluids, which causes increased blood flow, increased leakage of fluid and plasma proteins into the tissues, and other tissue reactions that helps to inactivate or immobilize the antigen. Inflammatory effects. Several complement products contribute to local inflammation. Next is alternative or properdin pathway. Unlike your classic pathway, it does not involve the immune complex, that is, it is independent of antibody. When we studied about classic pathway, we understood that antibody was needed for activation of the classic pathway, because after binding with the antibody only, proenzyme C1 was converted into enzyme C1 and cascade was started. Here we don't need antibody. C3 will be having an unstable thioester bond that will cleave C3 into C3a and C3b. Now C3b can bind to the surface of organism. So C3b on the surface of organism will bind to a plasma protein called factor B, and this binding will leads to exposure of a binding site for an enzyme called factor D. Now the factor D will come and bind to that binding site. Now this factor D will cleave factor B into factor BA and BB. So the cascade continues and ultimately leading to the formation of a membrane attack complex that leads to lysis of the pathogen. So in your classic pathway also you studied at the end there was a formation of membrane attack complex that caused lysis of the organism. Like that here also at the end there is formation of a membrane attack complex that leads to lysis of the infectious agent. This pathway is triggered by bacterial toxins, viruses, fungus, IgA, IgD, cobra venom, tumor cells, etc. Since we are studying about the actions of antibodies, that is indirect actions of antibodies via complements, here this pathway doesn't require antibodies, so you don't have to study more about this pathway. Just a few points are enough about alternative or properdin pathway. Next is mannose binding lectin pathway. Mannose binding lectin is a plasma protein which binds with mannose residues or mannose groups on the surface of bacteria and initiate the pathway. This pathway is activated in the absence of antibody. So it is independent of antibody. When mannose binding lectin binds to the mannose residues on the surface of bacteria, that results in recruitment of MASP, that is mannose binding lectin associated serum proteins into that site. Then mannose binding lectin and MASP becomes a complex. This complex activates complement component C4 and C2, and this activated C4 and C2 forms C3 convertase complex, and this C3 convertase complex cleave C3 into C3a and C3b. Rest is same as your classic pathway, and like your alternative or properdin pathway, your mannose binding lectin pathway also results in the formation of a membrane attack complex that leads to lysis of the infectious agent. So with this, we come to an end of the discussion on humoral immunity. So if you get a question, that write a short note on humoral immunity. Like I said, for cell mediated immunity, you should start with definition of immunity. Then you have to write the classification, that immunity is classified into innate and acquired. Acquired is further classified into cell mediated and humoral. Then you have to start mechanism of humoral immunity. Then you have to write the mechanism, that how these B cells are activated, then the role of helper T cells in activation of B cells, up to formation of plasma cells and antibody formation. Then you have to write the actions of antibody, that is direct and indirect. You have to mention all the direct mechanism. Then you have to write indirect action, where you have to discuss the 3 pathways, where you have to discuss the classic pathway in detail, writing that cascade of reaction, and you have to mention a few points about alternative or properdin pathway and mannose binding lectin pathway. Since we are discussing the indirect actions of antibodies, you don't have to discuss the mannose binding lectin pathway and properdin pathway in detail. You have to just write a few points about those 2 pathways. I hope it is clear. Thank you.